Morning, everyone. I'm going to speak into the microphone. I hope it's not too loud. Can you hear me all right? Yeah? All right, fantastic. Well, thank you so much, everyone, and thanks for making time to come today. My name is Fatima Nasrallah. I'm a researcher here and associate professor at the Queensland Brain Institute. And I'm also leading the Queensland Brain Injury Research Collective, which I'll talk to you a bit more about in this 5-minute introduction. So I hope I'll stay on time. First, I'd just like to open up by acknowledging the traditional owners of the lands on which we meet. We pay our respects to their ancestors, past, present, and their descendants who continue cultural and spiritual connections to country. So to give you a bit of a background, the Queensland Brain Injury Research Collaborative is a forum which was funded 2 years ago by the Vice-Chancellor at the University of Queensland. The idea behind the collaborative was to bring people together from all across Queensland in the brain injury space with the hope that we could form a research-focused network that can be driven by patient care needs and by clinicians and form a multidisciplinary network of people who can work together with the hope of really breaking down the silos and forming a one-team Queensland for brain injury. So this was support that we now have partnered with the National Injury Insurance Scheme Agency. They have been great supporters for myself and for the University of Queensland. The idea is that this partnership with the National Injury Insurance Scheme will allow us to expand QBIC for the next 3 years, so up until 2027. So they have been really great in that sense. So really our main goal for this is to link clinicians with industry partners, with community, allow for better outcomes for patients through the integration of consumer co-design, but also with that clinician's perspective into what we do. So I'll basically just first talk about introducing some of the researchers and clinicians that are going to be speaking today. We have a wide range, so I won't go into the details, but the people who will be speaking are perspectives from the research and the clinical aspects that are happening in the brain injury space, especially stroke. Given that this is Stroke Week, we're very privileged to have them here today to speak about the sort of what's been happening in this space. And we don't want to use this as just a one-day forum. We're hoping that this is going to be our first step for bringing people on board and expanding this network. So there will be a lot more that you'll hear from us, and that's why we're going to have the networking at the end, just to make sure that people can talk. It'll be great for you to spread the word around the Queensland Brain Injury Research Collaborative. Within this collaborative, we have 5 portfolios. We have stroke, we've got concussion, we've got moderate to severe traumatic brain injury. Innovation technology sort of spans these 3, and then we've got health economics and health services. And so the idea is that all these can come together to really provide that strong foundation for moving forward. We also want to share a lot of the work that's happening in Showcase. So outside at morning tea, you'll see the QBIC table where you'll get some brochures and some information more about that. But we also have Queensland Ballet joining us today. They provide services for brain injury patients, and we'll have a demo from them as well, which would be quite very interesting. And we're hoping to expand and work with them moving forward. And so I'd like to now really, and just one more thing. At the end also, there will be at the QBIC table some surveys. So we'd be really, really happy if you could provide some feedback on the day, but also provide some insights into where you'd like to see a collaborative like this moving forward. What would you like to see? What are the, what can we deliver for patients in terms of community reintegration, community engagement? So we'd really, really love to hear your feedback at the end. And so now I'd like to introduce our lead for the stroke portfolio that's within QBIC, Professor Dave Coupland. He's also the director of the Aphasia Centre for Research in Stroke. So thank you so much for being here today, Dave. Really appreciate it. So thank you so much. We look forward to talking to you today a lot more.
Thanks very much, Fatima, and thanks for the opportunity to speak with you all today. Thanks for coming in, and thanks to those online as well. So as Fatima said, I'm the director of the Queensland Aphasia Research Centre. I'm also at STARS Hospital, so I'm the co-director of education and research at STARS as well. And today I'm going to be briefly talking about language disorders that occur after stroke. So, the word aphasia encapsulates language disorders that occur after stroke, but in the general population, knowledge of what aphasia is, about 3% of the general population know what aphasia means. So, while this is very underdone in terms of common knowledge in the public, aphasia after stroke is incredibly common. So it's more common than Parkinson's, cerebral palsy, and multiple sclerosis. And what it involves is a language impairment and a communication disability. So it can affect any of these modalities. So it can be the ability to speak, the ability to understand what's being said, the ability to read and to write. And it's actually hard for us to, because we often take communication for granted, it's hard for us to truly appreciate how difficult this makes life. But if you think about this morning when you got up, how much did you communicate? So on your phone, when you talk to someone, when you got here in the car, when you got the bus, we communicate so frequently, and it's how we make connections, it's our identity, and it's how we develop relationships, and we have work as well. And so when someone has aphasia, it can be terrifying and sometimes devastating. So the most insight most of us have into this is those times when we can't find the right word. So something's on the tip of your tongue, but you can't quite get it. So then imagine that's all the time. So it can be incredibly frustrating, and it can lead to social isolation. So if you look at people with major health and medical conditions who rate their quality of life, aphasia has the most negative impact on quality of life compared to all major health conditions. We also know that the incidence of depression is significantly greater in those who have aphasia and have a stroke compared to the general population, and also those who've had a stroke without aphasia. So outcomes for people with aphasia are overall poor. So there's increased costs for rehabilitation, increased adverse events, increased time required for treatment, increased mental health and social isolation challenges, and increased stress for caregivers and loved ones. So at the moment, the outcomes for people with post-stroke aphasia are unacceptably poor, but there is hope. So we've been, I think, progressing in this field to develop new treatments that can improve outcomes, and also researching to understand the why. So why do people with aphasia recover in certain ways? Some people will recover almost completely. Some people, it will be a long journey and they'll need a lot of treatment. So some of the key challenges we've been looking at, and I'll be particularly focusing here on some of the work from the Queensland Aphasia Research Centre, is around inadequate access to services. So people with aphasia not being able to access the treatment that they require when they require it and in the amount that they require. There's been great progress in terms of trials showing evidence that aphasia treatment can work, it can be effective, but there's, as with most things, a very large gap between those research findings and what's happening in practice, where a lot of this research for what we know works hasn't been translated into practice. And finally, we have a large gap when we think about predicting outcomes. So for people with aphasia and their family members, often the number one question early on is, will I get better and when? And what do I need to do to get better? We still have a lot of work to actually give accurate information about this. So that's a third area that we've been looking at. So this is the team at the Queensland Aphasia Research Centre. So we're based out at the Stars Hospital on the Hurston precinct. And we're a UQ centre, but we're in partnership with Metro North and STARS. So one of the things that we've been developing over the last 10 years is new ways to deliver the type and amount of therapy that people with aphasia need. So based on the evidence that we know, can we actually get this delivered and what are the outcomes? So we've developed the CHAT program, so comprehensive high dose aphasia treatment. This program is 50 hours of treatment because we know people with aphasia often don't receive enough treatment for them to make gains that are meaningful. It's comprehensive, so it goes across the impairment and the function and the participation in communication. And it has different modalities, so it includes individual work, work with family members, education and computer-based work as well. So this has been delivered over the last 3 years at the STARS Hospital in a research partnership. And now it's really exciting that the CHAT program's now become business as usual at the STARS Hospital. So there's dedicated speech pathologists and allied health professionals and assistants who deliver it now. So over the last few years, we've been collecting some research data looking at the outcomes of the program. And what we've found is that it significantly improves outcomes in terms of how effective the person with aphasia is at communicating. So this is either through measures of their severity of their language impairment, or as seen here by measures from their partner of how effective they are at communicating. And importantly, this is also maintained at 3 months follow-up. And this has been a massive challenge for our area, is that often treatments have a significant benefit if you test people right after the treatment, but if you come back 3 months later, often those gains are not sustained. So what we think is happening here is there's not sufficient amount of therapy and there's not sufficient support for maintenance. We've also shown that doing the CHAT program also increases people's communication confidence. And also, if you ask the person with aphasia about their communication-related quality of life, this also improves. Because of some of the challenges with accessing such a program, so it's intensive, it's 3 days a week for 8 weeks, so 50 hours of therapy over 8 weeks. And this can be a challenge for people to come into the hospital and access it. So people with aphasia often have additional limitations in terms of physical mobility as well, or the ability to drive. So we've now been translating the CHAT program into an online version. And so this has all the elements of the CHAT program, but it's now delivered in the home. And our preliminary findings are that we get equivalent results for this program as well. At the centre, we also have the Aphasia Tech Hub. And so this is, again, to enable increased access for people with aphasia to the treatments that they need. The idea of the Tech Hub is it's kind of like those Apple Genius bars, but without the annoying people, and it actually supports you. So in this, it's a trial service we set up at the STARS Hospital where you can get consultations, you can get individual treatment plans that go from what are your goals to what type of technology might help with that. And we also have increasing capacity and training and education for clinicians, and also resourcing around how people can navigate this area of technology where there are many, many apps developed, and a large majority of them are not great, if I could put it politely. So not all the apps have evidence, but people are spending money on them. So we need to help patients and family members and clinicians navigate this new space. The Tech Hub has also been involved in then developing new tech solutions for people with aphasia, and critically involving people with aphasia at the start of this process. So co-designing from the initial thoughts around what would help through to the development of programs. So in getting clinicians, people with aphasia, students, designers, working together on how do you design an app for people with aphasia so that it's something that they will want to take on. And we've also had some aspects of technology that we've been developing based on the ideas of people with aphasia. So this is Gopal. He reported to us that he would often be told that he was making progress, but he didn't see it himself. And so that was a problem for him with motivation and attending and maintaining therapy. So we're now, through some funding, developing an app that then provides this kind of monitoring and feedback for the person and their family members on progress, on what the goals are, and then mapping how communication is working in real life. So is the person going out more? Are they communicating more in different contexts? And can we then use automated assessments of speech to get detection of whether there are changes in the quality and the content of communication? So the other area that I mentioned before was around the need to develop better clinical predictors of aphasia recovery and also treatment response. So often, currently, for clinicians, when they're developing a treatment program, it's based primarily on behavioural assessments. But we still don't know, so this is based on a profile of someone doing a language battery. We then think they might need a particular type of treatment. We don't know how much treatment, and we don't know how targeted that treatment should be. So this behaviour is arising because of the nature of the lesion, the size and the location of the lesion. But currently we're not able to use this information to actually work out what treatment should be delivered or what the outcome will be. So this is an example of the variation in outcome that we see in people with aphasia. So it's, at the present, it's highly variable and very difficult to predict. So what we've been working through is, are there brain-based measures that can help us predict outcome and whether someone will respond to a treatment? This includes some measures of brain frailty, so things that might have been present before the stroke, so measures of white matter intensity. Also then measures of the stroke, so the size and the location of the lesion. This is still largely one of the better predictors in terms of overall outcome, but it hasn't captured everything in terms of what you would need to tell a person with aphasia and their family members about what's going to happen. So then we've been using measures of functional activity within the brain's language networks and domain general networks, and also measures of connectivity with white matter. So disconnection within the language network is one of the things that appears to be predicting at least some of how much someone will recover and how they'll respond to a treatment. We're then looking at how those predictors relate to different elements of treatment. So the type of treatment that a person is given, how intense the treatment is. So we've looked at this where there's a large movement of people thinking massed practice or highly intensive treatment was best. But actually when you directly compare something that might be 50 hours over 3 weeks versus 50 hours over 8 weeks, there's some benefits for spreading out some of this treatment. This most likely relates to how the brain consolidates and relearns. And also understanding the dose or the amount of treatment that's required for every individual, for every type of treatment. This is something where we currently understand the vast majority of people with aphasia do not receive sufficient therapy. They often require a lot more than is currently available. And so we then need to think about how we can address that in a clinically meaningful way. So our next steps, we want to further develop these predictors of recovery and treatment response. We need to improve the way that we're measuring both outcomes and these factors that drive recovery. We're further developing these interventions. So we're rolling out, looking at the implementation of the chat program across a number of sites in Queensland, New South Wales and South Australia at the moment. And then how do we then support self-management for people with aphasia once they've finished some of these treatments? So it's another piece of work that we're working on. And in doing this, what we're hoping to do is to then be preventing some of the negative consequences and outcomes for people with aphasia. So a big thank you to the large team at the Queensland Aphasia Research Centre and all of our collaborators, particularly at the STARS Hospital. And there'll be a card table, I want to say, outside with some materials about how you might get involved in some of the work we're doing. Thank you.
Thank you so much, Dave. Just to move on to our next speaker, we've got Associate Professor Alan Pegner, who's from the School of Psychology, who's going to be talking to us today about how brain injury affects awareness and self-awareness. So it'd be great to have you here, Alan, and thank you so much for taking the time.
Hi, everyone. Yeah, I'm Alan Pegner, as the name is indicated here. I'm a researcher and a clinician in neuropsychology. I work at the School of Psychology. And one of the fields that I'm particularly interested in is that of awareness. And I'd like to give you a little bit of insight into this. Probably lots of you already know about this, but nevertheless, and then tell you just very briefly about some of the type of research that I'm currently doing. So the first question, before we start off, obviously, awareness is a term that can encompass many different things, right? So the first question that one would want to ask is, what do you mean by awareness when I'm talking about awareness? And obviously, there are things like awareness that is loss of consciousness, self-conscious, awareness of one's own existence, if you like. Someone in a coma will no longer have any awareness, any consciousness of themselves. That's not the type of awareness that I'm interested in. One of the surprising things that can occur following stroke, following brain damage, is, on the other hand, the loss of perceptual awareness. So what is perceptual awareness? Perceptual awareness is, for example, everyone might have moments where they lack awareness perceptually, right? You might be talking to someone and you will not notice something occurring in your visual periphery, or you might not notice something happening in the background. Now, interestingly, there are a number of things that can occur following stroke where this type of perceptual awareness is lost. Yes. So within this realm of awareness, of self-awareness, there are two kinds of things that can occur. On one hand, you can have a situation where a person will not be aware of the deficit that has occurred, that has risen following the stroke or the brain lesion. And there are some examples of this. There are many. I've just given two here. Things such as what we call anosognosia as neuropsychologists, which is a lack of awareness of a deficit. For example, a person may lose any sense of awareness of space, for example. They might lose the ability to orient their attention to one side of space and totally ignore anything that's occurring. Generally, it's on the left side of space, following stroke or damage on the right, in the right hemisphere. But there are other things that can occur, things we talk about, I mentioned in passing, somatoparaphrenia. That is something that also, again, is related to this loss of any awareness of part of one's body following stroke. And in some cases, it can go as far as to lead to somatoparaphrenia, which is the impression that the body part belongs to someone else. So a person, for example, in one case, a patient I once saw who was convinced, who had lost any awareness of the left part of her body and was convinced that when I asked her what this hand that she couldn't feel, who it belonged to, she said, well, I can't feel it, so it probably belongs to you. And as I was on the other side, she then concluded that it must have belonged to her husband, who was standing in the room further up. So this total loss of awareness and reconstruction of reality almost that is associated with this loss of awareness. Now that is the loss of awareness of the absence of awareness of a difficulty that has occurred following brain damage. But there's a converse situation that I'm particularly interested in. This converse situation is when people lose an ability, but they still have residual ability, but they're no longer aware of it. And that is something that is extremely interesting because first of all, it tells us a lot about how the brain is working, and it also might help in terms of rehabilitation. Now, there are examples of this. For example, following damage to the visual areas of the brain, one will lose vision on the opposite side of the visual field of space, but there might still be some processing ability that remains, even though the person is not aware of it. For example, someone with hemianopia who has no awareness of what's going on in the left side of their visual field, they might respond to something that approaches them in a threatening way. For example, an object that's approaching rapidly, they might readjust their posture, or they might turn around and raise a hand, even though all the time they are not, during that period, they're totally unaware of what's happening on the left. All right? Another example, yet another example that was captured once on video, a person who had no awareness of anything on the left side of space. Someone placed a bottle of water on the left side, asked them if they saw the bottle of water. The patient looks around on the right, is not aware of what's going on the left, says, no, I cannot see a bottle of water on this table. 10 minutes later, the patient asks for a glass of water, saying they're thirsty. And the examiner said, well, yes, certainly, go ahead, help yourself. And they turned around and grabbed the bottle on the left side of space. That's indicating that despite the fact they're not consciously aware of things on the left, there may be some amount of processing that's going on. It's not reaching awareness, but there is some processing going on. And that is one of the particularly interesting things I find, once again, both because it tells us about how the brain works and it gives us insights into perhaps how we could move forward in terms of rehabilitation. And I want to give you one example of a patient who became quite well known following the study, this study that we carried out, just to illustrate my point. This patient was, whose initials are TN, had suffered 2 consecutive strokes. You can see here his MRI several years after the stroke. These posterior regions of the brain where there's no longer any tissue illustrate the loss of visual cortex. He was unable to see anything. An ERP, electrical response, in response to visual stimulation, was totally flat. No information was being processed. No visual information was being processed by these posterior visual regions. Now, despite the fact that he would need help to move around, he indicated that he couldn't see, was unable to see. One chance occurrence demonstrated that there was, in fact, some processing. And this happened in a very straightforward way. I one day walked into the room where he was sitting in a seat. He looked up towards the door as I was entering, and I smiled at him without saying anything. And he, realizing obviously wasn't the right thing to do because he couldn't see me, but before I had time to say anything, he smiled in response. And his expression was exactly the same, what you would expect from someone who was sighted, which was quite surprising. I initially even thought that he had recovered entirely. I told him, well, it looks like what happened over the weekend, now you can see again. He told me no, but I thought I was under the impression that you must have smiled. Now, that obviously is quite strange. How can he be, what does that mean? So I actually tested this and presented lots of photographs of smiling or happy, smiling or sad faces, smiling or fearful faces, and so on a screen, and asked him to guess what the emotion was. He told me there was no point because he couldn't see. But I asked him to play along anyway and to guess. And it turned out that he was guessing above chance, even though he thought his level of awareness was insufficient for him to respond properly. And when we scanned him while presenting these emotional faces, we found that there was activity in an area of the brain called the amygdala, which is generally involved in emotion processing. So information, despite the fact that he had lost his visual processing regions, we can see that there were other areas of the brain that were actually receiving this information and that were allowing things to be processed, this information to be processed, although he was not unaware of it. Another chance occurrence, even more striking, we were testing him in a university in the Netherlands. As you can see, here he is, the gentleman with his white stick that he used to move around. Again, many years later, he was still blind, still couldn't see. But as we were walking through a hallway, he had his hand on my shoulder as I was guiding him to the cafeteria. And one of our collaborators, late Professor Weiskrantz here on the left, noticed that this patient had readjusted to avoid an obstacle, even though, once again, he's blind. As you saw, there's no visual cortex. So we decided to try and establish to what extent he could process information and use it to move around. I'll just show you the video. We took his white stick away, worried him a little bit. He was really not keen on doing this. And we told him, how about walking straight ahead and try and avoid obstacles? We just dumped things on the floor. This was... Yeah, I'm a lot younger on this video. So, again, asking him to go straight on and avoid the obstacles. And as you can see, again, he says he can't see, he's blind, he needs a white stick, but then when he's forced to try and move around, somehow he manages to find his way around obstacles. And we were absolutely surprised. It was really extreme. This was really totally unexpected. But we ran several studies afterwards, again, scanning him while we presented moving objects. And we found that different parts of his brain continued to respond to things like moving objects. Again, it doesn't reach awareness, but there is some processing going on through parallel pathways. So why is this important to examine and to look into this? Well, of course, first of all, as far as I'm concerned, we still, and it's been mentioned before, there's still a lot we need to know about how the brain works, and studying situations such as this, conditions such as this, demonstrate or help us understand a little bit more how the brain works. And of course, it has also use for rehabilitation, because one of the persons on the video, Professor Arash Sari, who works in Aberdeen, for example, has developed computer programs where he uses this ability to guess above chance as a way to help people recover a significant amount of conscious vision. So using this and encouraging people to guess whether or not things are occurring progressively have led to improvement in visual function. So yes, I'm currently working in that area, exploring again awareness of perceptual deficits following brain injury. And I think I'll stop there because I don't want to go over time. There are other people who have to present. So thanks for your attention.
Thank you very much, Alan. That was amazing and very inspiring, actually. We're very lucky today to have Queensland Ballet for brain injury with us. They've been an organisation which we've been introduced to through the National Injury Insurance Scheme Agency, but we're very keen to work with moving forward. And they're going to give us a demo today, and Laura Bailey's just going to give a bit of background about the program. Mimi. Yeah, so please go ahead. Thank you.
All right. Thank you so much, Fatima. So yes, my name is Mimi Van Buren. I'm the Community and Dance Health Manager at Queensland Valley, and this is Laura Bailey, my colleague and our Dance Health Specialist. So we're both really thrilled to be here with you today to represent Queensland Valley and the Van Norton Lee Community Health Institute to showcase our Ballet for Brain Injury program. We are both truly passionate about sharing the health benefits of dance and making dance accessible for everyone in our communities. Our Ballet for Brain Injury program began as a 10-week pilot program in 2021 in collaboration with Hopkins Centre at the Griffith University, Queensland Conservatorium Research Centre and Citrine Sun Entertainment. And since then, it's developed into a tailored program that we run weekly dance classes every Saturday at our home at the Thomas Dixon Centre. And we really welcome anyone living with a brain injury or anyone post-stroke to experience the benefits of health for dance and to really join us in a welcoming and uplifting community where the focus is really to spark joy and to just really have fun and enjoy. Our participants have shared that our inclusive, creative and adaptive approach to our classes has helped them in areas such as improved balance, movement and coordination, improvements in flexibility, memory, creative expression, social confidence and overall well-being. All right, so we don't want to talk for too long because we want to showcase what we do. And the important thing is that you will be doing it with us, as we have a golden rule in our studios that if you're in the room, you are actively participating, because the best way to learn is to do and to experience. So with that in mind, Laura is going to take you through some typical exercises that you could expect to enjoy in our weekly classes. And we really encourage everyone online to join in as well. Don't hide behind your screens. So we're going to take it away.
Awesome. All right. Thanks, Mimi. And thank you so much for having us. It's just such a privilege to be able to share some of the work that we do. And we have a participant from our Ballet for Brain Injuries class in the audience. So welcome and thanks for coming along. Margot, if you want to give us a wave. You can. Hi, Margot. Oh, so good. Yeah, it's an amazing program. And we're also out at Stars Hospital. We go there after this and we do some great work with some of the patients, the inpatients who are in their acute stages of, various different brain injuries. So yeah, we're going to start out. There's just 2 exercises today that we're going to explore. The first one is it sort of focuses on rhythm matching and a little bit of coordination. So we're going to have some fun and you can see how you go. The main thing is we're going to bring some awareness to our feet and our hands. So if you're sitting in a chair today, I encourage you to sit up straight. That's it. Just like a ballet dancer today. And we're going to have some fun. All right. So this is a really great exercise. It's sort of like a bit of a warm up, but also a challenge for a lot of our participants. It's utilizing obviously rhythm and dynamic, but also both sides of the body. And we like to get things moving at the Queensland Ballet. So thanks for joining in with us and we'll see how we go. All right, here we go. All right. Okay, we're just going to start out with some nice easy taps. That's it, team. Looking good. We're already a quarter ballet. Lovely. So good. All right, we're going to take it into our hands now. A little bit of a clap in a circular motion. Here we go. And it goes up. Nice. And down. Good. Once more. Good. We might try the other way. Reversing the circle. Good. All right. Bring it back to the center and back to the hands. Good. We're going to add a little bit into our feet now. So the toes are going to lift. Very good. 2 toes. We're going to keep that going and then alternate right and left, one side and the other. Good. Keep the hands going. If you want to change to just one, you can. That's it. Lovely. All right. Now we're going to take just some heel taps. Keep the hands tapping on the knees. Good. That's it. Good work. And really try and lift those heels. Well done. All right. We're going to take a little side to the side It's like windscreen washers. Nice. And we've got those toes going, hopefully, the same way as the hands. Good, very good. All right, now we're taking it into our heels. So the heels. And that's it. Perhaps just the lower part of the hand is helping drive. Good, that's it. Well done, everyone. All right, this is a little tricky. It's a bit like a shuffle. We're going to take a shuffle with our hand. That's it, on our leg. And then we're going to try that with our leg. Let's just bring the pace up a bit so we're in time with the music. Very good. I think I've got a few soloists in the audience. Let's get back into time with the quarter ballet, please. All right, let's see if we can do that with our leg now. Just one leg to start. We go out, in, out, in. It's a shuffle, like a tap dance shuffle. Good. Oh, we've got a few dancers in the room. That's it. All right, good work. We're going to bring it back in and then just switch over to the other side. Here we go. Good. And out, in, nice. All right, that's it. Let's go 4 more. Lovely. Now here's the fun part. We're going to take our hands together, feet come apart. Then we're going to swap. It's hands apart, feet together, change. Good. Out and change. Well done, everyone. We've got it. Do you think you could speed that up? I think we can. Wow. Don't think too much. That's it. And if you get it wrong, there's no right or wrong. Just keep going. Oh, try not to reverse it. And yes, keep going. Yes. All right. Let's just shake it out. Wow. Good. We're going to reach right up to the ceiling. Bring it in. And make a really big pose to finish. Wow. Well done. So good. So good. Oh, fantastic. Who would have thought such a simple movement could become so tricky so fast? That's it. So great. All right. So that's just one example of some great work we do in our classes. And another sort of area that we explore at the Queensland Ballet is we do use a lot of expression and gesture. And definitely when you're in the company and dancing in any performance, we don't always use our voice. In fact, we don't really use voice at all. And this sort of gesturing movement is how ballet dancers express a lot of what they're trying to say. So today we're going to learn a few gestures from a very famous ballet called Swan Lake. And this is going to be a little sequence that also helps with memory. So we're going to start off with a sequence that is where the prince meets the swan queen. And there is this movement which is you. Yes. So we're reaching out and we're saying you. That's it. You fly away. No more. That's it. Very good. Lovely. We've got some fabulous gesture in this room today. Well done. Okay. So you, good, fly away, no more. And then the next sequence is you, here, why. Good. And then the swan queen goes, I am the queen of the swans. Yes, that's right. And so we have this sequence here with a number of gestures, and that's sort of the story in itself is starting to evolve, the Swan Lake story, but also we have some movement here where if people can't speak, they can share with this gesture and they can express themselves in another way and another form. All right, so we're going to have a go at this in a sequence, and then we're going to have a go at some beautiful Swan Lake dancing today. All right, so let's have a try. And good. We might start with some warm up arms, and I'm going to have a quick drink. All right. Yes, good.
Warm up. Swan arm.
All right. Good. Sorry, I went running this morning. Okay. Thanks, Mimi. Good. And we reach and come down. All right. Ready to share in our gestures. As the prince, you fly away no more. Good. That's it. And again, you come here. Why? Good. And then the Swan Queen goes, I am the queen, good, of the swans. And yes, that's it. We've got a beautiful, beautiful quarter ballet here of swans. How fantastic. All right, let's try that again, those gestures. As the prince, you fly away no more. Good. And then you here. Why? And she comes in and she says, I am the queen, yes, of the swans. Good, beautiful, that's it. And we might just try a little bit more here. We're going to make this gesture of the lake. That's it. Good, using two hands here. Fabulous. And perhaps the lake is a little bit forward and back. Yes, in its flowing and rhythm. Good, that's it. Lovely. All right, we're going to try again, just with small circles. That's it, of our wrists. And you can be a prince or a queen today, whatever you like. We're going to take one arm up and down. Yes, and up and down. Beautiful arms today, everyone. Who would have thought you would have learnt some Swan Lake repertoire today? Fantastic. All right, good. Let's try two big wings. And as you're lengthening up, you're going to try and sit up tall and grow up tall. Yes, we're going to finish with a very beautiful extension out to the audience. And gestures are usually large and they're quite big because there's always thousands of people in these audiences that we dance to. So we're going to finish with a big reach up. We're looking up at the top tier. We're going to do a lovely bow. Yes, thanking everyone today. We're going to go out to the side. Thank everyone towards that right, good side of the auditorium, all the way out to the left. A lovely bow. It's a thank you. And just down to the orchestra pit, because we can't forget the orchestra, thanking them for playing for us. And the other side of the orchestra pit. Thank you. And a beautiful finishing pose of your choice. Anything you like. Oh, wow. Thank you so much. Very good. So good. Thank you. So that's another exercise, and that incorporates some repertoire from the company, but it's a really good one that really shares the opportunity to express yourself as well. And we find that in some of our classes, the joy is coming in and being a dancer for an hour or two, not being a patient. And I think that's really important for folks who come, and I'm sure Margot will attest to that. Being able to be a dancer and share that experience is something that we feel is of great benefit. So yeah, thanks for joining in. Yes, feel free to come and ask us any questions outside. Thank you so much.
Thank you very much, Mimi and Laura. That was great. And it sort of broke our somewhat very bare scientific presentations. So really appreciate it. And hopefully people can catch up with you at the end of this talks. So now we're going to have, oh, sorry, please go ahead. Yes, please go ahead.
And the prognosis was really grim. It's been grim all the way along. And even when you start to improve, the brain injury community here finds it so hard to see the improvement. I'm trying to work out why that's the case, that people just don't want to acknowledge that the stuff that this institute knows, why that can't take place in real life here. I don't get it at all.
Okay, I'm going to have to pass this question off to somebody who is a clinician in the room. Dave? Andrew would have been good, but I think he's just stepped out.
I'll look. So briefly, I think this has been a common issue that we've identified as well, where there's almost a divide or a lack of keeping up with what research is telling us about recovery and the brain mechanisms that drive that and prognoses, and what advice you might get as to what your potential to recover is. And I think this is something where we need research to be translated into, so for people who are giving that information clinically, to be more aware of actually where research is up to now in understanding that there is a lot of recovery that is available. That message hasn't got through to a lot of, not a lot, some clinicians. And I think part of that is potentially a belief that things like rehabilitation are not as effective as we now know it is. So sometimes the message is you've got a very limited amount of time to recover, and after that you won't. But that's actually not taking into account what we know now is if you get enough therapy of the right type and amount, you can continue to recover. So it's a translation of what we now know is possible into the messages people are getting clinically. Yep. And sorry, it remains a big issue that we're trying to resolve. Yep.
It is changing the better.
That's true. So I think part of what I'm keen to do is then how do we influence people who are training as clinicians about understanding prognostic information and how to deliver it and what is accurate and how important it is, both giving hope, because the message that you give at that point can have a huge impact on motivation. Yep.
Thank you. I hope that's addressed your question. And I do think that research moving forward has a lot to do with co-design with patients and participants who are affected by specific conditions. So the fact that there's becoming a lot more of that and in really integrating them into the science that we do is a huge step forward. Yeah, thank you. Okay, so now we've got Dr. Margaret Moore, our very own research scientist here at QBI. She's going to be talking to us about vision and special attention post-stroke. So thank you so much, Margaret.
Hi, everyone. I don't think mine is going to be as fun as that was, but I'll try my best. So I am a researcher here at the Queensland Brain Institute, and I do a lot of work with stroke, and particularly how the brain changes after stroke. So a lot of my stuff is targeted towards trying to understand what is going wrong in specific conditions so that eventually we can do a better job of designing better treatments for those conditions. And one of the conditions I work with the most is called neglect. So neglect is a really common post-stroke cognitive impairment in which stroke survivors have trouble seeing one side of space. And so importantly, this isn't a vision problem. Your vision is fine, but your attention just isn't letting you pay attention to things on one side of space. What this can look like in the real world is things like not being able to find things on one side of space, not eating food off of one side of your plate, not shaving or putting makeup on your face, or becoming a really dangerous driver because you're no longer doing a good job of looking at both places when you're making turns. What this looks like in the clinic is these are some of our standard tests that we run. So on the left is going to be a figure copy task in which someone is shown this picture of a little house in the sun and asked to copy it. And someone with neglect will often not copy one side. This other one is a cancellation task, which is one of my favorites, which we give to stroke survivors. And we ask, can you go ahead and cross off all the hearts on screen? And as you can kind of see here, this person has only crossed off hearts on one side. And that's a classic sign of neglect. And I think neglect really, really matters because first off, neglect has a really strong impact on people's ability to live normal lives. If you have neglect, you have trouble interacting with your environment, performing daily tasks and things like driving. This is from a study we did a while ago looking at how just interviews from stroke survivors, looking at how severity of neglect predicted all these problems. So people with more severe neglect reported having more trouble finding items, having more trouble paying attention to their environments and their bodies, and also being able to use their arms to perform tasks that they needed to do. Neglect is also really important because neglect is often a very happy story. About 80% of neglect recovers within the first 6 months following stroke. But in cases where it doesn't recover, there aren't that many well-accepted therapies to help these people. And so this is a chart from a study we did a while ago looking at what recovers and what doesn't. And everyone in blue recovered on their own with no therapy. So that's great. But our question now is, what do we do about the people in the red? And this little guy wearing glasses over here this is prism adaptation, which is a really common treatment for neglect. But again, as David mentioned, it's one of those things that might have short-term benefits, but there's very little research done on what long-term benefits are. And my idea is I think this problem could be because therapies like this are targeting symptoms rather than treating the underlying causes of neglect. And that's what my research is about. So my research goals are first off to understand how having a stroke changes how the brain works on a very basic level, and then trying to use these changes as a way to understand what's going wrong in people who have specific problems. And so this is all very theoretical, very taken away from the actual day-to-day life of a patient. But my goal is to hopefully that this will be something, an important first step to people doing better rehabilitation eventually. So what I do is I use cognitive neuroscience approaches, but at the moment is a lot of EEG. So what that is it's a cap we put on your brain or your head. If you want to see it later, I've got one outside. And we record the electrical signals coming off of your brain. And the electrical signals are caused by your brain doing things. And that's how we measure activity. So what you do is you come in, you look at the screen, and I measure your brain activity. After that, we take your activity, which is these little squiggles of electricity over time, and we do a lot of math. And what this math mainly is it's looking for patterns within your brain activity that tell us what was on screen. So for example, we can use machine learning to say this specific pattern of brain injury means that you were looking at a parrot, or this specific pattern means there was a pineapple on screen, and we can track that information over time. So this is what these kind of charts end up looking like. The green is kind of the level of information that's in your brain, and it's across time. And however high it is above being flat, that means how much information was there. And this is complicated, but it's really cool in terms of stroke because what we can do is we can measure information from both sides of your brain separately. And in people with stroke, one of your side of your brain has been affected by the stroke, one hasn't. And so effectively what we can do is we can start comparing how is information being processed in the side of your brain that's been damaged by a stroke and the side of the brain that's normal anyways, and we can start seeing these patterns come out. And what I'm looking for in my research is if I can find a pattern that helps separate people with neglect from people without neglect. And these are just some examples of what that pattern could look like. So the first case over on that side would be a sign that there's kind of been a loss of attentional balance between the hemispheres, because normally you see big differences in decoding between the hemispheres. But if you don't see it, maybe something's going wrong with just attention as a whole. In the second case over here, this is an example where you see the first green peak that's big and then it goes away really quickly. This would mean that your brain is processing information when it first comes in, but then it's being lost very, very quickly. So this would let me know when in the information processing pipeline something is starting to go wrong. This last case is similar to that, but at a later stage. And that we would be able to say, okay, your early visual areas have processed everything, but at some point it's dropping off between when you become aware of it. And so these are just some kind of things. What we're looking for is when is information lost when people aren't seeing things. I do a lot of other research. A lot of it has to do with lesion mapping. And what that means is when you've had a stroke, looking at the areas of the brain that's been damaged and comparing that over very, very large groups of people, so usually 2,000 or so, to try to figure out what those brain areas were doing in the first place. And you can get really complicated with the methods of this and look at things like brain networks and disconnection. So there's a lot of work that can be done just from that data. And one important thing you're going to hear from me a lot today is that I am always recruiting research volunteers. So if you have any interest in getting involved in my research, I'm recruiting both people who've had a stroke and people who haven't had a stroke to take part in exactly this kind of thing. And if you have any questions about what that would be like, you can come talk to me afterwards. This is the team. So this is our big lab group. And these are the people who've helped support our research. So thank you for listening.
Very much, Margaret. Very well delivered. Appreciate it. Just the next on our list, Dr. Matilda Balby is a professor here at the Queensland Brain Institute, and she also works on stroke. So she'll be talking to us about understanding how stroke damages the brain. Thanks so much, Matilda.
Thanks, Fatima, and good morning, everyone. So today I'm going to give you a quick overview of which kind of cells are damaged after stroke. And this is a bit of a different perspective than what you have heard so far. But before jumping into it, I'd like to start by talking about how the brain keeps its function in a physiological conditions. So the brain has a very high basal metabolic demand. It uses up to 20% of the body energy in terms of oxygen consumption, while accounting for only 2% of the body weight. The brain has a very limited capability of storing energy substrates, and this makes the brain a very hungry organ. And to satisfy this hunger, the brain needs a constant delivery of oxygen and nutrients from the bloodstream. Therefore, a very tight regulation of cerebral blood flow is needed, as any imbalance very quickly affects brain function. This intricate relationship between brain activity and blood flow requires the action of a plurality of cells. Very often we hear of neurons. These cells right here, and we all focus on neurons, especially when talking of diseases, but there are multiple cells that play a role, and these cells are all composing what we call the neurovascular unit. There are cells such as astrocytes, for example, these big cells right here, and have multiple functions. They are even involved in clearing the brain from waste. We're going to talk about astrocytes in a second. I just want to tell you more about microglia right here. This is another example of cells, and those are the brain resident immune cells. Now, as you can see from this schematic, all these cells are either directly or indirectly connected with each other and connected to the blood vessels right here. And this communication, this interaction is essential for physiological neurovascular coupling, which is the mechanisms that guarantee a coordinated response between neuronal activity and the blood flow. The idea here is that to a local increase in the brain activity, it needs to correspond an increase in the blood flow. When a stroke occurs, we have basically an obstruction. Blood, either to the brain or within the brain, cannot flow, and this leads to brain damage. Now, blood flow is interrupted either by a clot, and in this case we speak of an ischemic stroke, or a vessel bursts and leaks, and in this case we speak of an hemorrhagic stroke. In my lab, we mainly focus on ischemic strokes, and we work on animal models because this allows us to measure things that we could not measure in human patients. And we use brain stimulation, and I will get to that in a second. So when we look at the brain after the occurrence of a stroke, we can see that the area that surrounds the occlusion, the brain tissue becomes hypoperfused. And over time, we see a different pattern. We can observe a different pattern. It's like observing two different zones. The area that is closest to the occlusion becomes an ischemic core, and the surrounding area, the periphery, is what we call the penumbra. Now, in the core, cell death occurs within minutes. In the surrounding area, instead, the tissue is hypoperfused, but is still viable. This is what we call at-risk area. And that's because over the course of the time following the occlusion, there is an increased likelihood that this tissue will die and form the final ischemic lesion. However, if blood flow is promptly restored, this area may still be rescued. And this is why in our research we target this area for intervention. As I mentioned before, we work in animal models, and that's because we can we can measure things that we couldn't in humans. And in our lab, we use brain stimulation, so we measure the responses of neurons, vessels, and other components of the neurovascular unit. The video that I'm showing you here shows you how we can use optogenetics, which is a technique that allows us to shine light on the brain and activate these specific channels here. These channels are strategically located on our target cells, and in this way we can click on and off. We can turn their activity on and turn their activity off so that we can control what they will do after stroke and during our intervention. Using this approach, we have stimulated these little cells here. Those are inhibitory neurons, and those are part of the neurovascular unit. And with this approach, we have activated these neurons at a specific frequency immediately after stroke, and we have observed a neuroprotective effect. What we saw specifically was an increase in cerebral blood flow, a reduction in the occurrence of spreading depolarizing waves, which are events known to occur after stroke, and the occurrence of these events is correlated with a worse outcome after stroke. And we also saw an improvement in motor performance. Later on, we also saw that our stimulation was able to rescue the brain ability to have connections within and across different brain regions, a process called the functional synaptic plasticity. More recently, we have been interested in a different cell type, the responses of a different cell type, astrocytes. As I mentioned before, astrocytes play an important role in clearing the brain, getting rid of all the waste that the brain produces. Astrocytes have many other functions. They are not just involved in neurovascular coupling, they are involved in inflammatory responses, vascular repair, synaptic transmission regulation, all mechanisms that guarantee brain homeostasis. Now, as you can see in this beautiful picture here, this is how an astrocyte looks like. All its branches, and you will see why it's located. So astrocytes are strategically located between blood vessels, which are right here, and their processes reach blood vessels, and they also reach neurons. And that's because they have a very important role in communicating, in helping communication between neurons, blood vessels, so that the coupling I mentioned before can occur normally. After stroke, things change. This is how the tissue of an healthy subject looks like. And this is what happens after stroke. In this case, we were staining for astrocytes. So this little disrupted pattern that you see here is all astrocytes. So what Victor is doing in the lab, Victor is a PhD student in the lab, is trying to understand how we can modulate this communication and use the techniques that we have in the lab to improve recovery of function after stroke. Another thing that we do in the lab is to we stimulate the brain, and at the same time we can record the brain activity and behavior. In this specific example, the mouse is doing a behavioral task with the affected paw. He's reaching for a water droplet, and that mimics what will happen during rehabilitation. At the same time, we can record the brain activity and see what's happening in the brain. And the idea here is that we can adjust the parameters of stimulation based on the ongoing brain activity and ongoing behavior of the single mouse. And this brings me to the mission of the lab, which is to develop an ideal future therapy that will involve behavioral tasks for patients that induce coherent activity in the brain and uses artificial intelligence-driven customized stimulation to enhance physiological and emotional recovery. And this is what we try to do. This is what we are aiming to do in the lab. So with this, I'd like to thank people in the lab for showing up every day and being excited about the science that we do and what they can contribute to stroke research. I'd like to thank the donors and the fundings that is supporting our lab. A special thanks goes to the Brazel Family Foundation. They allowed me to be here today. And all the support that we get over time. Before thanking you for the attention, I actually would like to invite you to come to the lab. If you're interested in what we're doing, please reach out, send us an email, and we are always very welcome to show you what is that we do in our labs. It's extremely exciting for us to show what is it we do and how every day we try to improve lives from our little spot. Thank you all for the attention.
Thank you very much, Matilde. And just now we are privileged to have Ron Walsh, who'd like to talk about his lived experience with stroke. We really thank you for your time today, Ron, and appreciate the information that you're going to provide us, our audience here. So thank you.
Thank you. Hi, I'm very much the amateur here. I'm not a professional speaker and I'm not a medical person. I'm just someone who had a ischemic stroke last year, 5 days after my 72nd birthday, and it scared the living daylight out of me. It really did. I did not expect to have a stroke at all. If you had asked me the week before, you know anyone that's likely to have a stroke, I wouldn't have picked me, that's for sure. I was in pretty good shape. I was pretty healthy. I'd stopped drinking alcohol for about 3 months. One of my 6 sisters said to me, "That's why you had a stroke, you idiot." That's not true. Anyway, it was an ischemic stroke from a cholesterol build-up in my artery in my neck, and I had a left anterior artery endarterectomy in my neck since, and I'm now on medication to reduce cholesterol, and it's working well. The impact of the stroke on me has been amazingly insignificant. I don't think I've had much of a negative impact from the stroke. My wife says my memory is not so good, but she's a lovely person. What's her name again? Denise, that's right. But apart from that, I mean, I go to a gym occasionally and there's a guy goes there in a wheelchair and his name's Andrew, lovely guy. And I've been going there for a few years and I've never said to him, I didn't go to the gym after my stroke. And I went down finally and Andrew said, "Ron, where have you been? Haven't seen you for a while." And I told him and he said, "A stroke? You've got to be kidding me." And we got talking about it and I said, "Andrew, I've never asked you, how did you end up in the wheelchair?" And he said, "I had a stroke." He said, "I had a stroke 7 years ago," and he's paralysed almost completely down the left side. So I feel extremely fortunate to have come through the way I did. Why? I have no idea. Your researchers looking into the effects of stroke on the brain, I would love to know, what those effects. I've been on the website. I'm now a volunteer speaker for the Stroke Foundation on recognising when someone is having a stroke and then what to do about it. And I find that very rewarding and important. it's a message that needs to be put out there, not just to older people, but a lot of young people have strokes too. Far too young. So it's an important message. So I'm going to, they asked me to speak for 5 minutes, but I think you're running late. Is that right? I'm going to be out here if anyone wants to ask questions, and I'm certainly willing to volunteer for any research that any of you might be looking into, if I can help in any way. Okay, thank you. Enough?
I've just asked, does anybody have any questions for Ron? Oh, we do have a question, Ron.
Yes. Thank you for sharing that experience with us. Very interesting. And I was wondering, when you had your stroke, what were the first things you noticed? What made you realise something wasn't right?
Yeah, sure. As I said, it was a few days after my birthday, and it was actually my daughter's birthday. And we were at an Indooroopilly hotel, actually, and I had a drink after 3 months without any, so I felt pretty good. And the right side of my face just went numb. It just actually sagged, and oops, and I thought, you know, what on earth is that? And there was a medical professional in the room. Actually, there's a lady that runs medical suite of offices, and she came over and she said, "Ron, are you okay?" And I went to talk to her, and the right side of my mouth drooped, and I couldn't get the words out. They stumbled, and she said, "Sit down. I think you're having a stroke. I'll call an ambulance." But they never did. They never did call the ambulance. But they kept talking to me. They kept asking me, "Are you okay? Are you okay?" And I eventually said, "No, I think I'm fine." And I think it passed. The stroke passed, and my wife took me home to get changed to go to the hospital. We got home, and I said, "No, I'm all right." And I laid down on the bed, and I slept for 12 hours. And then on the Sunday, I went online and booked in to see my doctor. Still didn't go to the hospital. I didn't get to the hospital until Tuesday. So I was extremely lucky that there wasn't more damage. But yeah, it was the right side of the face and the speech that was all stumbled. So very scary, very frightening.
And as you said, I guess the key message there is if you have any of those sort of known signs or symptoms like the weakness or the numbness or slurring of speech and things, to sort of act pretty quickly.
Yes.
And it's no big deal to go to a hospital and have yourself checked out.
No, or call or call an ambulance. That's the main thing. Is when they said to me, "We're going to call the ambulance," they should have done that. In fact, the least competent brain in that hotel, probably in all of Indooroopilly at that time, was mine, because I just had a stroke. And they didn't call the ambulance, but I was lucky. I was lucky to get away with being relatively sane at this point, I think. Thank you.
Thank you very much, Ron. Thank you for sharing your experience, Ron. So I think it's one of the main things that keeps driving the research that we do, listening to these stories and making sure that we can sort of spread hope across patients and improve outcomes for patients. Look, I wanted to thank you all for being here today. As you can see, QBI has a very strong platform in terms of stroke research. We're very thankful to all the donors who donate to QBI to support the work that's being done at the basic level, but also at that translational level. And do you want me to say something, Matt? Yes. Oh, okay. Matilda's talking to that. Okay. Do you want to do that before I wrap up? Sorry, Matilda's just going to announce one of the top-up awards that's been given to our participants. Oops, come on. Here we go.
So it's my pleasure to introduce the winner of the QBI Stroke Research HDR Top-Up Award for 2024, which is Victor Matsuda, which is a PhD student in my laboratory. So Victor is working on, as I mentioned before, on astrocytes, and he's trying to understand how we can modulate the astrocytes and improve their communication with the neurons and blood vessels after stroke. He just started. just come close. He just started and we were very happy to see that many donors were actually supporting PhD students like Victor. This is extremely important for us because we give the opportunity to Victor to leave his PhD this year with a bit more of support, and this is great. So I'd like to thank, I'm not going to thank each single person, but all of them for their support. Thank you so much for this, and well done, Victor.
Thank you very much.
Thank you very much, Fatima, for facilitating this, and thanks to all our speakers. I think we've had a really good indication of the range of research that's going on and the implications of it for improving outcomes across many aspects of how a stroke can impact on a person. And please keep in contact with the Cubic. We're keen to further develop our relationships across, so researchers, clinicians, community, consumers, because I think that's how we'll make sure that there's this translation of research into practice and better outcomes. So we have some demonstrations booths outside in that direction, where there will be some information. You can sign up if you're interested in being a research participant. Also get some further information about some of the groups, both here and at STARS, and work that's going on that is looking across the range from mechanisms and how strokes impact the brain through to better treatments. So we'd encourage you to take some time. There'll be some volunteers there you can speak to. So there's also the Stroke Foundation has some information here as well. And thanks very much, Ron, for participating and giving your story. And this is also an opportunity just to meet with people, have a chat, network, which is what this collaboration is all about. So thank you.